Okay, so let's talk about Toxoplasma gondii. So it has definitive and intermediate host, right? So in definitive host there is sexual cycle, so that is cat or man, which is also man is its dead end host or accidental host, because once it infects the man there is no continuation of life cycle. Intermediate hosts are rodents and birds. So now let's talk about its life cycle. So starting from infective stage, which is sporulated oocyst, and there are two. All right. So this is in soil. All right, and this is taken by rodents like rat, and in rat it forms tissue cyst. And these small units are called Bradyzoites. All right. So this mouse is taken by cat. All right, and then in cat's muscle it is formed into Bradyzoites in singular forms. Now this Bradyzoites form merozoite, and this merozoite can form again tissue cyst. All right, and if it undergoes gametogony, then it forms macrocyst and microcyst. All right, so these two goes under sexual cycle and forms oocyst, and this oocyst again forms into sporulated oocyst, and this life cycle continues. But if by any sorts of reason this sporulated cyst is taken by human, then in human it is formed into Bradyzoites, and this Bradyzoite is formed into Tachyzoites, and this Tachyzoite is distributed in different tissues like brain, muscle, and by death of man this cycle just finishes. So in human this cycle get its stop, and humans are accidental host. So that's why it is called man is called dead end host of Toxoplasma gondii, or accidental host, because they are not supposed to be infected by the Toxoplasma gondii. Now if we talk about the laboratory diagnosis, okay, so I have done one mistake. So this is not microcyst, this is microgamete, and this is also macrogamete. So macro and microgamete. So this cycle takes place in intestine. So this is called enteric cycle. enteric cycle because it takes place in intestine, and this is called exoenteric cycle. All right, and one more thing, this Bradyzoites forms sporozoites, means sporulation takes place, and then after infective intestinal mucosa, this forms Tachyzoites and then goes in different parts of our body, mainly the brain, which causes various problems. So now let's talk about the laboratory diagnosis of Toxoplasma gondii. So first thing is microscopy. In microscopy, there is peripheral blood smear, in which we can found like this. So these are Tachyzoites. All right, and if we cut tissue cyst, then we can find this is Bradyzoite. All right, so in tissue cyst we can find Bradyzoite. In peripheral blood smear we can find Tachyzoites. Now moreover we can do ELISA, PCR, IFA, that is immunofluorescence assay, all right. So these are the different laboratory diagnosis which can be conducted to check for the Bradyzoite of Toxoplasma gondii. So this is okay. So some extra test can be Sabine Feldman test. So this is the gold standard test for Toxoplasmosis. All right, and in terms of CNS involvement, MRI can be done. All right, so this is all about the laboratory diagnosis of Toxoplasma gondii.